Welcome back to another video tutorial on capsule staining by Indian ink method. Okay, so let's get started. Capsule stain. Now what is capsule? So I think as we all know, so it is the outermost layer of the bacteria and it is gelatinous layer. Okay, and they are made up of polysaccharides or polypeptides. So as you can see in the picture over here, this colored layer which you can find around the bacteria, so this is called as capsule. Okay, and then what is the function of the capsule? So this capsule is the one which provides pathogenicity to the bacteria. Okay, and apart from that, so they also protect the bacteria. Now for example, when this bacteria enters the host cell, so the host will show some immune responses, right? So all that immune responses will be protected by this capsule. Okay, see for example like phagocytosis, desiccation, host immune defense. So all these immune responses will be protected by the capsule and it makes the bacteria safe. And not all the bacteria will contain capsule. Okay, there are only some bacteria which contains capsule. Example is Klebsiella pneumonia and Streptococcus pneumonia. Okay, the next thing is why do we require a special staining technique for capsule? Is because capsules are non-ionic. Okay, so what is the meaning of non-ionic? Non-ionic means they lack electric charge. Because they lack electric charge, you cannot stain them easily with stains. Okay, you cannot do gram staining for them easily. We require a special staining technique. So it is difficult to stain with basic stains also, acidic stains also. So we do a special staining technique for capsules. Okay, so yeah. Okay, so next coming to the principle of capsule stain. Now as we know, capsule is non-ionic. Okay, and they cannot take up any stain. So may it be acidic stain or may it be basic stain, it cannot take up any stain. And if you try to stain them also, they remain colorless. They do not take and they remain colorless. Okay, so as a result, what we do is we stain the background here and we stain the bacterial cell. But the capsule will not take up any stain and they appear colorless. Okay, see now. What all stains we use here is a negative stain, which is Indian ink or nigrosin, colors the background. So as you can see in the picture over here, this is a picture, and we use negative stain, that is Indian ink or nigrosin, that completely stains the background. Background becomes completely dark. And we use another dye, another basic dye, it may be crystal violet or saffronin, and that stains the bacterial cell as you can see here. So this is a bacterial cell, they appear purple in color and they stain the bacterial cell. Okay, so whereas if the capsule is present in the slide, so the capsule appears colorless as you can see in the picture over here because they cannot take up any stain, so they remain as a clear halo here. See around the bacteria you can find this clear halo, colorless clear halo. This is the capsule and you can visualize them very clearly because the background is completely dark and the bacterial cell is also stained and the capsule will very clearly be visualized as a clear halo. Okay, so next coming to the Indian ink method. So yes, we also have other method called Antony's method for capsule staining. So here in this PPT we will study Indian ink method. So in Indian ink method basically we use two stains, okay, or two dyes. First one is crystal violet and second one is Indian ink. Okay, and if the capsule is present in the organism, you can find a clear halo or colorless region in the surrounding the bacterial cell. Okay, so as you can see in the picture over here, so this dark background, no, so this dark background is by the color of the Indian ink. Okay, and this here the bacterial cell is stained. So usually the bacterial cell will stain purple, so because of the stain crystal violet. Okay, so usually the bacterial cells will be negatively charged, hence they take crystal violet and they appear purple in color. And if the capsule is present, you can find a colorless clear halo surrounding the bacterial cell. Okay, so usually we do this for Cryptococcus. So this is Indian ink method. Okay, coming to the detailed procedure now. So in the procedure, what we actually do is we take a clean glass slide. Okay, and then in one end of the glass slide we place a drop of Indian ink. We add a drop of Indian ink, and then in the next step you will take a inoculation loop, and then you will flame that. Okay, you will flame that, so you will remove all the organisms present, and then in the clean flamed slide you will just take the culture tube or the plate. Okay, and you will take the organism. You will take the organism from the culture plate or tube, and then you will put it onto the Indian ink. So make sure that you should spread the organism evenly. Okay, otherwise so there will be clumps, and when there is clumping you cannot see the organism clearly. Spread that evenly. So after spreading, what you can do is, so after spreading, if you want an even smear throughout the slide, what can be done is you can take another clean glass slide like this. So place on this, only the edge has to be placed on this glass slide, and you can drag in such a way that the smear now completely spreads the glass slide. Okay, that is the Indian ink along with the organism will be completely spread throughout the slide. So this can also be done. You can take the clean glass slide, keep it in such a way, just the edge has to be kept here. It can be dragged so that the smear completely spreads the glass slide. Okay, so once this is done, now you will allow it to air dry for about 5 to 7 minutes. So we will not heat or we will not blot dry because the capsule may melt. Okay. Yeah, in the next step, now after air drying, in this step what you will do, you will flood the uh, slide with crystal violet. Okay, so after flooding it with crystal violet, you will keep it for 1 minute. After 1 minute, so you can water wash it. So you should be very gentle while water washing because excess water may remove the capsule, and that's the reason you should be very gentle in washing while washing. And then after that, in the last step, you will air dry for few minutes. Again, you will not blot. It's very compulsory that you should not blot. So blotting will remove the bacteria from the slide or distort the capsule. Hence, blotting or heating is not recommended here. And then so now the slide is ready, so you can observe them under oil immersion microscope. So as I told, so the background will be completely dark in color, and the organism will be stained purple in color, and if there is capsule you can find a clear halo. Okay, colorless halo, so which you can see around the organism. So for the result, see, look for the purple cells surrounded by a clear halo. So on a dark background, the halo is a capsule. If halo is present, yes, that is a capsule. You may need to decrease the amount of light in order to make the capsule easier to see. Yes, since it is dark background, light has to be decreased so that you can have a good visualization.